Hello, our team is Tralero Tralala. And we are Tristan, Ju Chan, and Jasmine. This is our presentation for Biology 101, completed on October 17th. Today, we will be talking about osmosis and diffusion in Elodea leaves. The purpose of this experiment was to observe how Elodea leaf cells respond to different concentrations of sucrose solution in order to estimate the molar concentration of the Elodea leaf. Specifically, we wanted to see how water moves into or out of the cells by osmosis, depending on the outside solution. Our hypothesis was, if Elodea cells are placed in distilled water, they will become turgid as water enters the cells. In contrast, if placed in concentrated sucrose solution, the cells will plasmolyze as water exits the cell. At the molar concentration that matches the inside of the cell, there should be no net movement of water and no visible change in cell shape. To carry out the experiment, we prepared 6 slides by adding Elodea leaves to each. Each slide was exposed to a different solution, distilled water, and sucrose solutions of 0.2, 0.3, 0.4, 0.5, and 0.6 molar. Key steps included cleaning the microscope lenses and labeling each slide, placing a small Elodea leaf on each slide, blotting excess water, then adding 50 microliters of the designated solution to each slide, placing a cover slip over each sample and waiting 5 minutes for the solution to act, observing each slide under 400 magnification and photographing the results. Here are the pictures and videos. Now, let's check out the results and observations. When observed under the microscope, in distilled water, the Elodea cells appear turgid, with chloroplasts pressed against the cell wall. In concentrated sucrose solutions, such as 0.5 and 0.6 molar, many cells showed plasmolysis. The cell membrane visibly pulled away from the cell wall, and chloroplasts clustered towards the center. In an isotonic solution, the Elodea leaf would remain in a normal state, because there is no net movement of water into or out of the cells. The cell membrane will stay close to the cell wall, and the cells will neither become turgid nor plasmolyzed. Here are labeled images from our lab experiment, showing the different cell appearances at each concentration. Now I'll explain what happened in terms of osmosis and diffusion. In this experiment, we saw clear differences when Elodea cells were placed in different solutions. In distilled water, the cells looked turgid. That's because distilled water is hypertonic compared to the inside of the Elodea cells. Water moved into the cells by osmosis, creating turgor pressure. This is the pressure of the cell contents pushing against the cell wall, and it keeps the cells firm. But in concentrated sucrose solutions, the environment was hypertonic. That means the solution outside the cells had a higher solute concentration than inside the cells. Water moved out of the cells, so the cell membrane shrank away from the cell wall. This is called plasmolysis, and the cells looked much smaller and less firm under the microscope. Somewhere in between, in an isotonic solution, there would be no net movement of water, so the Elodea cells wouldn't change shape much. This isotonic point is important because it reflects the actual molar concentration inside the cells. Let me define a few terms. A hypertonic solution is less concentrated than the cell's interior, so water enters the cell. A hypertonic solution is more concentrated, so water leaves the cell. An isotonic solution is equal, so there's no net water movement. Plasmolysis is when cells shrink because they lose water. Turgor pressure is what keeps plant cells firm, thanks to water pressure. For a real-world example, salt kills slugs by drawing water out of their bodies. Similarly, freshwater fish can't survive in salty water because osmosis would pull water out of their cells. So, our experiment clearly demonstrated osmosis and diffusion, and helped us estimate the molar concentration of Elodea cells by finding the solution where there was no net movement of water. In conclusion, this experiment confirmed that osmosis causes Elodea cells to swell in distilled water and shrink in concentrated solutions. Our hypothesis was supported by the observations. Based on the results, the molar concentration of the Elodea leaf appears to be between 0.3 and 0.4 molar, where cells were neither fully turgid nor fully plasmolysed. One experimental source of error could be varying thickness of leaves, impacting water movement. Some images were AI generated and selected from Google. Thank you.